Homo sapiens. How are you? Welcome back to studying microbiology, Prescott's microbiology. Today's topic is cell membrane, prokaryotic cell membrane. In reality, eukaryotic cell membranes and prokaryotic cell membranes are not that much different in their structures. Not that much, right? Like the basic component is the same. So this is how usually the prokaryotic cell structure looks like. Wait. Something's wrong. So this is how it generally looks like. In this topic we will be just covering what the cell membrane is, why does a cell membrane exist, like what are its function and how it is structured. These are the three main points we will be discussing. First, the name, the cell membrane or the plasma membrane. Membrane is the covering of something, right? The covering of something. And in this case, the covering is of the cell. In the case of the name of the plasma membrane, this is the covering of the plasma. Plasma is that liquid portion in the, or you can say the cytoplasm, but only removing the organelles, right? Cytoplasm is the plasma plus organelles. So this kind of protects that liquidy layer. The membrane's function is not just to protect the cell. It has a lot of functioning. If you remove the plasma membrane, or if you disrupts the or degrades the plasma membrane, the cell is done. And this is way more important in prokaryotic cells because prokaryote is a single cell. It doesn't combine with any other cells, right? And yeah, it doesn't combine with any other cells. So if the normal functioning of the cell membrane gets disrupted, so it won't function normally and it will destroy. And in the case of eukaryotic cells, it does function normally, but it does have other cells to protect. Like in prokaryotic cells, it is the very basic component to be studied, and it has to have the characteristics to protect the cell. So in eukaryotic cell, it has less functions than the prokaryotic cells, right? In prokaryotic cells, they starts from acquiring nutrients to eliminating the waste, and not just that they do in the respiration, the photosynthesis, even that functions are done by the cell membranes, right? So in this paragraph they said that they interact with the environment through the cell membrane because it does have proteins and lipids, and it acts as a signal receptor or as a signal transporter. In the same case, it is permeable, selectively permeable, mean it only allows certain molecules to enter and certain molecules to let out because of its nature, dual nature, because it is polar and it is non-polar at the same time. So the material that will be coming in and out will have to be passing through this, right? Now the plasma membrane, they are in this whole paragraph they are saying that it have a lot of roles. How much roles does it have? Like I said, it acts as a transporter system, and it is way more complex than it looks, because the transporter system, wait, so, this one is way more better. So because the transport system does not only include the proteins and the lipids or these kind of things, it also acts as signal transporter or the vesicle transporter. Now has protein secretion, hair, photosynthesis, respiration, metabolic processes. It also has the components to, you can say, to make the cell wall. The cell wall making factory is kind of plasma membrane. The things don't come from the inside of the cell. It is manufactured in the cell membrane, and then it is transported to make the cell wall, right? So cell membrane is important in all of the cells, but prokaryotic cells mostly depend on it, like more highly depend on the cell membrane. All of them do, but prokaryotes even more, right? So that's the kind of functions they do: selectively permeable, transporter, the protector, mechanical protector, chemical protector. of the cells, signal acceptor. Yeah, so these kind of things. Now the structure there written is the fluid mosaic model. From fluid it shows that it is fluid, but that doesn't mean that the layer moves around. It means that the material that is within the cell, embedded in the cell, moves around, right? And this is the most acceptable model. There are a lot of other models for the you can say cell membrane structure. That is, for example, lipid bilayer. Lipid bilayer is just a part of this fluid mosaic model, right? Because there are two layers of lipids, right? Now, what kind of lipids do we have in the cell membrane? We have to study that. Number one. The second is what kind of proteins are in the cell membrane. The third is what kind of fats or carbohydrates or lipids are in the cells, right? Now generally the structure is there are two layers of lipid bilayer, right? Now the lipid looks some kind of like this. It has a head and it has a tail. Lipid bilayer means one layer has a lot of these lipids, and the lipid looks like this: the phosphate head and the fatty acid tail. Now there is a structure problem, or you can say structure complexity, that makes the plasma membrane permeable. Permeable means allowing something, right? Permeativity, giving permission to something. Now the head looks like a phosphate. Oh, sorry, not look like a phosphate. The head is the water loving region, right? Wait, these ads distract me. Water loving region. The tail is the water hating region. That's why the area that is in contact with the environment and that is in contact with the water, or you can say plasma of the cell, cytoplasm, because it has high constituency of water, is the water loving site, or aka hydrophilic site. The one that is hidden from those water sites are the phobia, right? It don't like the water, so it is inside. Now if you look at the structure, why is it hydrophilic and why is this hydrophobic? It is because of their structure, chemical structure. This is, look at this. This is plus and this has phosphate group, right? This is known as ethanol amine. If you don't have, you want to go deep into that, just remember it has a phosphate group. That is, if you can see, it has a minus on this side and it has a plus on that side. That makes this structure polar. That means it will be attracted to water because it has the polarity in this. But if you look at the bottom structure, it doesn't show a positive and negative sign. It's just kind of neutral type, non-polar. It doesn't have polarity. And these are not so much shorter structure. The upper one is exactly this much structure, but the lower one can be longer and longer. Depends on R, how long the R is. R is the alkyl group, right? Alkane, you can say alkene, alkyl. It can be anything, right? And the R length is measured by how many carbon is in this R. Now this is just the structure. So this is how it is arranged, and they don't intersect with one another. There is a space between these two if you look at this, and within these spaces the proteins integrate themselves into it. Now how the protein integrate into them when they have dual nature, the polarity, hydrophobicity and the hydrophilicity, right? Now the protein has a positive and negative nature combined in them. Protein structures are complex. Now the area which is hydrophilic, the water loving, that is outside of the cell. Now if you look at this, you might think that, look, all proteins half come out of the membrane and half out of the membrane. There are some proteins that doesn't show out their head, you can say this. They have half and half structure. Like half of this, the whole what am I saying, hydrophobicity region. So it won't show this head out, and it will only show this tail out, that is hydrophilic. But in this case, these proteins are all the blue proteins. They all have on both sides the hydrophilic region, and in the middle part they have hydrophobic region. Look at how tangled and springly it made itself into the layer to fit itself in it. And if you look at the same length that is outside, they didn't spring themselves up because they are free, they love it, and they have more space in that. And these are just the two different types of the proteins. Ever heard of globular protein or the fibrous protein, right? They are different types, so don't mind why the one looks rounder and why the one looks lengthier, longer. Now, these proteins that are embedded in the lipid bilayer, no, like it's not fixed, but it can only be in this position. It cannot rotate, it cannot be reversed, overlapped, or anything, but they can move in this direction if they remain in this type of structure, right? Like this much area up out of this membrane and this much inside, they can move in this phase inside this whole lipid bilayer, right? As you can see, bodyguards that's moving across the whole layer, but they cannot come out of it, cannot come inside of it, and cannot change their position, not position, you can say angle in the cells, in the membrane. Now there are proteins. Now these are integral protein because they are integrated into the lipid bilayer. Now there are peripheral protein. Peripheral means, out on the outside, peripheral side. They are not embedded in the cell membrane. They are either attached to the integral proteins or just outside the proteins, right? Like there is no portion in this cell that is inside the, why am I saying cell again and again? There is no part in the protein that is inside the lipid bilayer. No, they could not rotate. They cannot rotate. Abdul Ali. They cannot rotate. They will be in the same state as they are right now. They can only move, you can say, forward and backward, side to side, but not flipping themselves up, right? So the other types of proteins that are in this lipid bilayer are these red types of protein. Now the opposite type of the peripheral protein must be the type that is completely within the lipid bilayer, and they are called as hapanoids, as they showed in from the red hopanoid, sorry, not hapanoid. And no, vibratory is a different thing, because vibratory means moving only from one point and from mean position, right? Like it has its range from moving, but no, these proteins can move all across the membrane, right? Not just on a fixed position. They can move wherever they want. Well, the vibration cannot be occurred in the whole area. It is fixed. Anyway, so these are the three types of proteins: the peripheral protein, the integral protein, and the hopanoid, right? These are the general categories of the protein. Now we have carbohydrates and the lipids. And what other things remains? Lipids, carbohydrates, no, nothing else remains, right? Now the carbohydrates don't usually, you can say, are separate. They are attached to either lipid or either protein, like either glycolipid or either glycoprotein. Mostly are in the form of glycolipid and glycoprotein, rather than being in oligosaccharide form or polysaccharide form, or even in monosaccharide form, right? They usually don't exist. They are more like this. And the glycolipids means, of course, the glyco means, glucose, carbohydrates. Lipid means lipids, fats, whatever. Now, the glycolipids or glycoproteins act as signal receptors. We usually say, right, that when someone attacks, they have to attach themselves to the receptor of the cells. Where are these receptors actually? So these are the things that are receptors, and they are not just receptors for outer material, foreign materials. They can be receptors for the nervous system signals, nerve signals. They can be for that as well. Like if the nerve signals it to not secrete the whatever kind of protein, not secrete them outside the cell, then they will tell the cell that, yeah, do not secrete, and of course the cell will stop secreting. The plasma membrane will understand that I do not have to do that. By the way, these integral proteins also act as channels. Some of them act as a channel, or most of them act as channels to allow ions to pass through them, proteins to pass through them, lipids, water, whatever to pass through them, right? So the channels are because of these integral proteins as well. Because these are channels, they have to be, both up and down to allow things to pass through. Now this is just the general idea of the lipid bilayer, right? Like there is not anything else that is basic in the structure. If we go deeper from that and other molecules, that will be specific for the all kind of cells. For example, bacterial membranes, archaeal membranes, eukaryotic, fungal membranes. By the way, cell walls are drastically different from one another, but the cell membranes are, kind of similar. Now, in this paragraph, they showed that how did we study this lipid bilayer structure. They said it is done by the transmission electron microscope. Transmission electron microscope means they studied the internal structures. Guys, you get the internal structure and the external structure, right? We study the scanning microscope, electron microscope, study the outer structure by collecting the reflected electrons, while this one collects the refracted electrons, right? And the lipid bilayer is just about 5 to 10 nanometer in thick. This is talking about, I think, prokaryotes. Well, of course, all the cell membranes, but if you look at from the prokaryotic perspective, prokaryotic cells are in micrometers. So you have to guess that the cell membrane is going to be even more, thinner than that. Or, of course, if you think about the length, the area, it is going to be larger. I think it is the biggest organelle in the cell because it covers the whole cell, exception to the vacuoles, right? Some vacuoles are way larger and thicker. Now, how did they know that these integral proteins are in the cell membrane? This is the freeze etching technique we studied in the previous lives. This is a technique where we freeze the cell and cut some part of it, even if it is cutting the middle part of the cell membrane, like cutting just one layer of the lipid bilayer. So you see these integral proteins and stuff like that. Yeah, and they are talking about the lipids. Amphipathic, there is a word used. Amphipathic means they are both hydrophobic and hydrophilic, both water-loving and non-water loving. That doesn't mean that they are sometimes water-loving and sometimes not water-loving. They are both at the same time. Half of the part is hydrophobic and half of the hydrophilic. Now, which one is common, do you think? Peripheral proteins or the integral proteins? Of course, the integral proteins are more common because they act as channels. Channels are the important working of the plasma membrane. So I think my topic on just the general cell membrane ends, and I won't be going further. Bacterial membranes and the archaeal membranes. Now they have different characteristics, right? Different characteristics. We will study this in detail with another live, like cholesterols. Like look at this. The structure already have things different because of its cholesterol there. And archaeal, look at these structures, ester bonding, stearic acid. Actually the fatty acid is different. Patanol, this is also the fatty acids are different. Yeah, the chain is different in these. So anyway, this will be discussed, in the next one, because I didn't want to confuse the people that just wanted to study the cell membrane structure. So anyway. this is the end, and yeah, bye. See you in the next one.